Hello and welcome to this week's What Were They Thinking? Among the range of news items we have for you this week is the rather amazing cost of tobacco products in Australia, at an average pack of cigarettes being nearly $50. A experimental treatment for spinal cord injury has about a 70% success rate. Why it is that explosives training in the military might well lead to leaky gut syndrome. A interesting approach to vaccinating individuals, but for a parasitic infection rather than your typical pathogens. A Hong Kong man hospitalized after being bitten by a monkey, and possibly starting the movie Contagion. A fossil explanation as to how ears and teeth developed. A publication by Kevin Falter, among others, on the molecular, biomechanical, and physiological aspects of grafting fruit trees. The overlap and similarity between urban sprawl and cancer, or as you may better know it, that scene from The Matrix. The development of levitating graphite plates for incredibly sensitive sensors, primarily for use in quantum mechanics, and a interesting take on the use of full stops and modern communication. The timestamps for these and other news items can be found in the description box below. First, we have what is arguably a similar example of how the cost of living just keeps going up, no matter who you are or what you do. In this case, though, it's on what could arguably be described as a luxury item, tobacco products, and specifically the example is to buy a single pack of cigarettes. In Australia, due to government taxation amongst other things, the average cost of a packet of cigarettes, that's between 20 and 25 individual cigarettes, will cost you more than $50. Allowing for the inflation, since the article was published, it's going to be closer to $55, if not $57. After all, 2023 and 2024 have been, a no small degree of increase in the inflation rate. This is down to the excise tax being applied, and all of the associated health measures that have been brought in. For example, plain label packaging, the terrifying eyeball, and so on. The rationale of this approach is rather straightforward, and that is, the more expensive a product is, the less appealing it is. Which is a great idea in theory. In practice, it doesn't exactly work. It's why drug addicts, although being addicted to an expensive product, still find ways to pay for it, and not necessarily legal ways either, and this is possibly what will happen with tobacco in Australia, which is a concern. Of course, this is, in theory, intended to help with all of the health issues associated with tobacco consumption, something that really is only going to be solved by stopping consuming tobacco, something New Zealand tried and has since rolled back. In other health-related news, we have the effect of fasting-style diets and changes in the human brain, which isn't that surprising. When you are going to be working on a system that has so many hormones related to the idea of eating, the moment you start fasting, you will stop having the same kind of impact on those hormones, leading to different signals going to the brain. This is very much a case of, no shit. But studies of what happens, and albeit a very small study, finds that although the individuals lost weight, about 16 pounds or 7.5 kilograms, which corresponded to roughly 8% of their body weight, the part of the brain that was being, affected by this appeared to be related, at least somewhat, to the microbes within their GI tract. The very oversimplified version is that the changes in the GI tract are known to be associated with changes in the brain, just not exactly how and for how long. The changes they did identify in this case via functional magnetic resonance imaging, or fMRIs, is in parts of the brain that are known to be involved in the regulation of appetite and addiction. Therefore, theoretically, it should, and understand theoretically is said for a very simple reason, it's a small cohort, a relatively uncontrolled study, and for a relatively limited period of time, may have implications for obesity. On the other hand, it may not have any kind of relevance. It needs a much larger study to be able to know more about that. In slightly more significant results, we have what can be best described as a controversial study. It involved implanting stem cells into the spinal cord of those who have been injured. Now, to be very clear about it, it's a phase 1 clinical trial, so it is small and relatively preliminary. The concepts are established, and generally speaking, animal models will have shown that everything works as intended, but translating from animal models to humans is never perfect, and that's what we've observed with this same study. Where 10 patients were given the stem cell treatment, only 7 had improved movement and sensation, which means a 70% success rate, which is, well, to be blunt about it, it's not great. It is true that 70% is much better than the typical 5% that you would see without this kind of treatment, but having 30% without an explainable lack of improvement is a problem. This is where phase 2 and 3 trials would theoretically have the place and role to play, and that's because phase 1 is mostly focused on establishing safety, and there doesn't appear to have been any notable adverse events that are going to deny this from proceeding further along the clinical trial pathway. In other clinically relevant news, we have a possible trigger for asthma. To be clear, we are talking about a relatively unnoticed and distinct trigger. That is distinct from things like a pollen and other similar, relatively benign allergens, which is what trigger asthma in many cases. Rather, what we're talking about is the mechanical and physiological process that leads to it occurring. If we were to compare this to a otherwise more easily understood scenario, what the researchers describe is that the area that makes up your airway becomes overcrowded with cells during a asthma attack, much the same way that a hallway full of people during a rush gets filled with people and they jostle around. In an asthma attack, these cells, due to the overcrowding, get killed, similar to how a stampede of people might lead to people being injured or killed. When this happens, it triggers inflammation, at least in the case of cells. For humans, it's panic. In order to compensate for this panic or inflammation, there's an increase in mucus secretion, and this then leads to increases to damage in the airway. This effectively creates a self-fulfilling cycle which continuously goes round and round, getting worse. The reason it gets worse is that, quite simply, the barriers that should be there to prevent the allergens that cause asthma get damaged further, meaning that the allergens are more easily able to trigger another asthma attack, which means the barriers get weaker, and so on. Why the research is useful is that the researchers propose targeting this mechanism to, at least in theory, and understand that this hasn't actually been tested in humans yet, prevent the exacerbation of the underlying mechanism by protecting that mucus layer and the barrier, or hopefully allowing it to recover after an asthma attack. You can prevent the damage from occurring and the escalation of the asthma attack over time. Of course, this has really only been established in an animal model and not humans, and as already stated, translating from animal models to human studies is, not particularly successful. In terms of success, it's a success of a kind is our next news item. How training in the military with explosives might be what's causing veterans to have leaky gut syndrome. Importantly, we're not talking about massive explosions like artillery shells. cannons, and so on. Rather, much smaller explosives, things like hand grenades, charge packs, and so on. Explosives that you wouldn't typically consider as being the reason why somebody would have effectively shell shock. The theory behind this is that because permeability of the GI tract, and, well, the intestines in particular, is controlled by neurons, there is a possibility that the relatively small-scale explosions from things like grenades could be decreasing cognitive function and thereby interfering with issues around control of the gut, leading to leakiness. This is, well, a nice theory, and it's not exactly well substantiated, to be honest. It involved about 30 participants, and they're all male, which isn't a massive problem for this particular kind of study. But 18 of them had mild traumatic brain injuries from blunt force trauma, which means that more than half of the individuals involved can't necessarily have the symptoms related back to their condition, which really does undermine the whole point of the study. Further to that, the research was primarily observational. Admittedly observational in terms of they had the individuals involved recreate the environment and situation of something like an explosion, exposure, and then subsequent blood tests for the next 16 or so hours. So, again, theoretically the study has meaning, but a whole bunch of the study participants already had other issues, and it's really hard to be able to correlate what they've found with what they're claiming to have found. Shifting from what is currently clinically important news to news that's more around diseases and pathologies. The first item being a new material that is basically a caltrop, but for viruses. It's a small, spiky ball intended to be able to burst virus particles, thereby preventing them from being able to either contaminate a surface or, more likely, and certainly more importantly, infect individuals. It's essentially a microscopic spiky ball, which you won't be surprised to learn, given how dangerous it sounds, was developed in Australia. The idea of this design, basically, is that they're able to impregnate material with it and create a surface finish that is fundamentally inhospitable to viruses, bacteria, and other microscopic organisms, with a roughly 96% effective rate of killing whatever comes into contact with it in terms of microscopic life forms, which would be perfectly fine for maintaining a relatively sterile environment. The only issue with this concept is just how hard it is to make the product in question. The entire technique used to create it requires a stream of ions to cut away thin parts of silicon wafers. Effectively, the same sort of technology scale that you would need for microchips. Admittedly, thankfully not as small as a microchip at roughly 290 nanometers, but still, this is at a ridiculously tiny scale. In other microbiology-related news, we have a strategy to deal with other pathogens that doesn't involve creating a spiky environment reminiscent of a Slaneshi cult party. Rather, we have what would probably make Papa Nurgle happy. Well, happy and then very sad. It's a vaccine strategy to deal with parasitic organisms, and in this case, we're talking about a particular kind of pathogen, Schistosoma, and these are a fairly common kind of parasitic organism. They cause about 250,000 deaths a year, and more than 500 million people worldwide are infected. This is why the vaccine idea is not only promising, but in some terms, incredibly important. Why this particular organism, let's say, is difficult to vaccinate against is the same reason that most organisms are hard to vaccinate against. It takes a lot of time and effort to find an antigen or the right kind of marker on it that will not only stimulate the immune system and stimulate it to an extent where it will actually have a memory of the organism, but do so in a way that it doesn't trigger on self-targets. For example, your own red blood cells. And therein lies the challenge. It's incredibly hard to do, it requires a lot of work, and therefore it's expensive. And, well, most people don't have the money to fund this, barring possibly Jeff Bezos and Bill Gates. And, well, Bill Gates is already funding all sorts of research elsewhere, likely not going to be getting himself involved with this immediately. That brings us to just how researchers are trying to tackle it without both the money of Gates or the intent of flying into space of Bezos. What they're doing is a similar, if not the same technology used for COVID-19. What they've done is take a whole bunch of bacteriophages, and with all these bacteriophages, they've put all of the known protein sequences from the Schistosoma worms into them individually. That is roughly 11,500 different genes responsible for making proteins. They've then tried to find out from the monkeys that were then infected with these bacteriophages which of them were expressed and, importantly, at what time. They found more than 99% of the sequences they used showed up in the plasma to some extent, which means these were probably some very sick monkeys. But, importantly, what they found was different genes were expressed at different times, therefore leading to different amounts of protein at different times. And this means that, theoretically, they can take this information and be able to figure out which proteins could be useful in which ways for a vaccine. In fact, they tried just this, admittedly not using a typical vaccine format, but just the protein gene candidates and then consequently the proteins themselves in a vaccine. The mice given this vaccine showed a significant reduction in the number of parasites, but it's worth us pointing out that not only was this a very crude form of immunization, but they weren't delivering it as a typical vaccine. There was a lack of things like adjuvants and more, all of which would drastically improve the performance of the vaccine. In similar news, we have a new antibiotic that could help with blood infections in particular. Sepsis is one of those things that you don't want to ever hear from your doctor. Being told, "I'm sorry to tell you, you have sepsis," is kind of like hearing, "I'm sorry to tell you, you have cancer." Admittedly, a cancer is worse, it's much harder to treat over the long term. However, over the short term, sepsis is infinitely more alarming. It is able to kill you in very short order. In terms of what causes it, there are several species of bacteria that are particularly likely to cause things like sepsis. And the challenge with these organisms, or more accurately bacteria, is that they are very hard to treat with antibiotics. Most of them are things that are gram-negative, and because they're gram-negative, half of the antibiotics that are available, if not more, don't work anymore. And because you don't have those antibiotics, the primary targets of the bacteria are much harder to get through. This leads us to what you do to treat these particular bacteria and this newest antibiotic. The antibiotic is looking at an enzyme within gram-negative bacteria, the gram-negative bacteria being those that are hardest to treat, and the enzyme within them, it's called LPXH. This enzyme is responsible for the lipid membrane that is around these bacteria, and that lipid membrane, if it's not working properly, it becomes a weakness that, well, is being exploited by this antibiotic. Ironically, the compound itself is called JEDI 1444, or Jedi 1444. The drug itself comes in a range of variations, and by modifying it, they're able to show an increase in efficacy, and it's an increase in efficacy after a single dose. It's worth us pointing out, in case you're getting excited about this, that there is a very good chance that this investigation won't lead to anywhere in the short term, and certainly not by itself. What is a similar dead end is the case study of a 37-year-old man from Hong Kong. He was infected with a virus after being bitten by a monkey. Yeah, this does sound like the start of the movie Contagion, the movie where an individual brings a monkey back from Africa and starts spreading a disease that causes an entire town to be put into lockdown. Sort of like COVID, but on a smaller scale. The other interesting thing here is that what he was infected with is the herpes virus simiae, or B virus. The Hong Kong man literally got monkey herpes by being bitten. The man in question can, to a certain extent, consider himself in an elite club, as he is one of about 50 people who have been diagnosed with this since 1932. Perhaps not the equivalent of the Mile High Club that he wanted to be in, but still, it's something to be proud of. Sort of. Maybe. Maybe not. Going from animals causing illness and bizarreness in humans, to humans causing weirdness and bizarreness in animals. Although, not perhaps what you're expecting. Somehow, researchers managed to get a mouse embryo to grow limbs where its genitals should be. There is a euphemism of calling the male external genitalia a third leg, but this wasn't a third leg, this was a fifth and sixth leg for the embryos. Where this gets kind of bizarre is that the way they did this was by turning on or off a single gene, the TGFBR1 gene. It's a receptor protein, and it, for lack of a better description, orients the mouse embryo in a particular way, saying this is the direction things should grow, and therefore, by measuring along these axes, you know that you should put the limbs here, 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 and here. But when you turn it off, you get two extra limbs that, well, aren't particularly helpful. In a rather creepy development on this, the otherwise developed limbs really didn't have much difference in the genes expressed compared to the typically found limbs. It's entirely possible that this might contribute to understanding just how it is that octopuses decide which of their legs will actually be a penis. Shifting from the very modern and disconcerting news to relatively ancient news along the same lines, but this time shifting to why and how we have our ears and teeth, not just where they are, but what they are. Admittedly, this is, amongst other things, based on fossil research, so it's somewhat more difficult to be able to get some of the details that would be nice to have, particularly around the structures in question, as all we can really identify are the bones that have remained, and this of course assumes that these are all of the bones that should be there, which does lead to the matter of how it is that Chinese, Australian and US researchers are collaborating. The difference between the species that were looked at in the fossil record shows that reptiles had one inner ear bone versus mammals that have three, which explains some of our greater hearing capacity and certainly the ability to better differentiate sounds the way we can. In a overlap between relatively recent news and somewhat older historical news, we have the possibility that the Sumatran tiger might well be alive in Indonesia. Well, specifically the Javan tiger within Indonesia, as it's a relative thereof, the Sumatran tiger, and could provide a way of, theoretically, conserving the population. However, there's one slight hitch to this. In 2008, the Javan tiger was declared extinct. You might wonder, why is it that you can conserve an extinct population? And in some ways, we're actually thinking along the lines of Jurassic Park. Not crazy dinosaurs and terribly designed and cursed parks. Rather, we're thinking more along the lines of evidence that the Javan tiger is still alive, and, if it is possible, the Sumatran tiger might be a functional surrogate to help the population. If it is alive, and that is a massive if. The entire premise of this is that, at least in 2019, a small sample of hair from a suspected tiger was found. In 2022, it was assessed, and it was found to have 97% similarity with the genetics of a museum specimen. Now, the important thing here is that this sample, along with other claimed sightings and dead livestock, indicates that there may be a wild population of Javan tigers in Indonesia. The challenge with this population is just the same as it is with koalas. It's incredibly small, and it would likely at this point be incredibly inbred and likely to have all kinds of issues around its health, let alone having almost no habitat to speak of, being likely to be shot if it hunts too much of the local agricultural animals, and if it doesn't hunt the animals, it might be hunting humans. This is why, if, and remembering it's a massive big if, the Javan tiger is alive, they would have to find a way to conserve it, and ideally conserve it, finding ways to keep the population alive and reduce the rate of inbreeding. Without this, well, the Javan tiger is pretty much up shit creek without a paddle. In news that's related to, well, more or less the opposite of conservation, we instead have the idea of taking existing trees, and likely exactly the same tree, cutting a scion from it and grafting it to a new tree, or more likely rootstock. And it's an article by Kevin Falter, who you may know as a ag science communicator, and it goes through the various aspects of grafting. Grafting itself can be an incredibly important and useful tool. For example, most nursery stock today is grafted. Sometimes it's done to dwarf the stock to prevent it from growing too big, a particularly useful phenomena for small yards. In other cases, it's to ensure that the plant can survive in certain soil or environmental conditions. In other cases, it allows for the provision of a relatively large tree fairly quickly, since the trees themselves may either have a very low growth rate, may have a very poor seed growth rate, or in other instances may just not want to grow because they're sterile. We won't go through all of the details, mostly owing to the fact that it's a rather extensive article, but it does cover things like the mechanisms of grafting, the various photohormones, the physiological, biochemical, and molecular mechanisms of incompatibility, whether or not you can predict that incompatibility, whether or not different rootstock is going to be important, the signaling mechanisms, and more. Going from agriculture to what could be best described as education, we have claims that math homework could be causing more harm than good, although this is an interesting argument to make. Australian researchers, so we have to wonder about whether or not the research is actually based on anything that can be, let's say, applied outside of Australia, has been working with various families and children from grade 3 or so. The children are around ages 8 or 9, which does also now raise questions about whether or not this is applicable to higher levels of education. If we were to take the premise, at least at its face, as being valid, The conclusions aren't so much. The first issue we have with it is that they're arguing that if it's too complex for students to complete even with parent support, they're wondering about why it was set as homework in the first place. But typically speaking, homework is set based on content covered in classes. Therefore, if the student doesn't understand what's being taught, this is generally a teaching issue, not a homework issue. Second, if it's for a child aged 8 or 9 and the parent can't understand how to do it, this is an indicator that the education system has already failed. Not those kids, but generations beforehand. The authors have also gone down the nonsensical pathway of arguing that because math can be difficult, that sometimes mothers, who generally are responsible for homework, may be inadvertently discouraging daughters from taking part. An utterly nonsensical position to take based on real-world data about different populations and what they do and don't do when it comes to which particular field they choose to pursue. Those countries with the most liberal possibilities of what you choose to take part in tend to, by virtue of simply having a choice to make, fall in particular ways. Those that are least liberal in what you can choose to do tend to find a more equitable split in subjects by the sexes. That is, in countries where you need to basically make money, people tend to go down the pathway of basically getting into the best paying job, no matter what sex you are. In a country where that's not so important, women tend to choose particular careers and men tend to choose others. It's not anything to do with what they were taught as children, because this has been accounted for in actual studies that don't spread nonsense like this does. Shifting to what you could describe as town planning or city builder simulator, it's an argument that those who are designing cities should, perhaps, look at the way malignant cancers grow and think about how they can apply this knowledge
I'd like to share a revelation that I've had during my time here. It came to me when I tried to classify your species. I realized that you're not actually mammals. Every mammal on this planet instinctively develops a natural equilibrium with the surrounding environment, but you humans do not. You move to an area and you multiply, and multiply until every natural resource is consumed. And the only way you can survive is to spread to another area. There is another organism on this planet that follows the same pattern. Do you know what it is? A virus. Human beings are a disease, a cancer of this planet. You are a plague, and we are the cure.
To the spread of the outer suburbia. Where things get interesting is looking at cities and their historical spread. For example, London over the last roughly 180 years. Over that time, there's a fairly well-established central nucleus. It is an area about 2 kilometers square or about 1 mile square. That very central core has, between 1831 and 2011, spread like crazy, with tendrils spreading out and creating the city that we know today, or at least we know as London. What is weird is how it spreads and the similarities to things like cancer. Specifically, the role that certain main lines have in creating the urban sprawl. Railway lines in particular, but also major roads. A feature remarkably similar to how arteries work for, cancerous growths. The idea from this is that by treating, the urban sprawl as being similar to cancer and using similar strategies to what is used to curtail and control the growth of cancer, it is possible to better control and develop cities. Admittedly, it's a theoretical solution because in practice, the theory and practice rarely translate so well. Something you would note if you ever watch any of the actual city planners playing things like a city builder. Shifting to similar population-related news, instead we have what could be kindly described as a rather hyperbolic claim that a woman involved in a gene tech company and fertility treatments could decide which babies are born, which, it's a bit much. In fact, it's a lot much. The fundamental idea is, rather than just blanketly practicing eugenics, the company involved is testing each IVF embryo and establishing which of them is least likely to develop complications later in life, and as a result, the most viable, both immediately viable and over lifetime viable embryo is selected for. It's a very big difference from choosing the best child based on various traits. This is entirely built on the premise of choosing the healthiest embryo now and in the future, which, compared to some of the allegories with 1930s painting school dropouts, isn't really a fair take. Fundamentally, all they're doing is the same idea that most IVF clinics will do anyway, except they scale up the screening of possible complications. Most IVF clinics will only screen for a small number of reasonably predictable and fairly common issues. This is just the same idea, but on a larger scale. As you'll read in the interview, the interviewer seems to have this particular issue grasping the idea of, rather than having somebody who can live a fulfilling life and be experiencing some hardship, you can make the hardship unnecessary by removing it from their life, thereby allowing them to have a more fulfilling and more complete life. Which is a drastic difference from the other argument they make of, but the person wouldn't exist. And as the interviewee points out, they would still exist, they would just be a different person. It's the same person in the same place, just it's a different individual. Effectively the multiverse. Obviously, the idea is not necessarily going to be for everybody, but the proposition and the underlying idea is fundamentally sound and very different how it gets presented. That is, this is more or less exactly the same as already occurs with IVF, it's just a greater degree of screening before the choice of embryo. Shifting from controversial news to something that's significantly less controversial, we have physics. Depending on how you experienced high school, physics may be seen as a great thing or a terrible thing. In this instance, it's better than average. It's a process that's been developed that allows for the capturing of Wigner crystals after about 90 years. The crystals aren't what you would typically think of as crystals. Rather, they are when electrons get forced together because of their own negative, let's say, repelling forces that would, by virtue of how they've all come together, get trapped in a nice, neat crystalline lattice. A distinction as separate from most of the rest of the universe, where we generally see electrons as being free to move around, particularly around their proton and neutron. This brings us back to observational evidence that the Wigner crystal actually exists. The researchers have been able to not just visualize that it exists, but visualize its formation, which is important because not only does it prove that it's a thing, but it also means that the properties that lead to its formation and make it happen and what happens when it's being made can also be confirmed. To be clear about this, the Wigner crystal is not a new idea. In fact, it's an idea going all the way back to 1934. And as you can imagine, given that we're talking about electrons, incredibly tiny things, it's no wonder it's taken so long to be able to not only visualize it, but effectively prove that it exists in such a direct manner. The technology to make this occur just hasn't existed before now. In other news, we do have something light-hearted, and it's more in the title than the actual content. The title reads, "Will any crap we put into graphene increase the electrocatalytic effect?" A rather straightforward and rather honest description of it, and the findings are, well, rather straightforward and to the point for the most part. The short answer is yes, they will. It's unlikely that graphene with bird droppings on it, as it's described, will be the next leading source of material, but nonetheless it does make an interesting point, and it's somewhat tongue-in-cheek. Shifting to news that's probably going to be more useful, we have levitating graphite, and the use that this plate of graphite might have for incredibly sensitive senses. The challenge with most levitating objects is that in order to make it happen, you tend to have to use various mechanisms that don't contribute very well to sensitive senses. The researchers from Japan not only are likely inspired in no small degree by anime, but also have found a way around this issue. Magnets. Powerful magnets. And by using these, they're able to make the graphene, or graphite more accurately, float, largely free of the forces of gravity. Shifting from what is very much current news to what is not going to be current for a very long time at this rate, but also finds its roots back in the 1950s. It's a solution to the Fermi paradox. The fundamental idea of the Fermi paradox is that the Earth itself is about 4.5 billion years old, and life is about 3.5 billion years old. Yet in all that time, humanity has, at least not to our knowledge, encountered any other life in the universe, a universe that is billions of years older. Why then is that happening? And the answer the article from the conversation comes to is the three-body problem. Or at least it's in the three-body problem, a Netflix series. The short version of this is that the universe is like a dark forest, and that everybody within that universe is a hunter. Every hunter is trying to avoid the other hunters lest they be killed. Therefore, the tactic is to hide and avoid each other as much as possible. Because once you encounter another organism, race, species, in short, extraterrestrials, humanities are pretty much doomed. Going from what is likely to be the doom of humanity at the hands of aliens to what is likely to be at least partly tongue-in-cheek, but also probably why humanity is doomed itself. Gen Z finds a period or full stop in communication to be passive-aggressive. The education system has failed us on so many levels, but this has certainly achieved one of the higher ranks amongst all of the failures of that education system. The final news item we have for you is all about the eclipse that occurred this week, and an article that the eclipse cost America almost $700 million in lost productivity. Yes, people took time to go outside and have a look at the phenomena that won't occur for 2.5, 3 decades, something like that in the future? Yes, that wasted 2.5 minutes to see the awesome astronomical event has cost America nearly $700 million, but likely that same phenomena has led to a huge increase in the various social media companies from them being able to sell the user data of those who have been sharing their half-assed images of said eclipse. There are a range of issues with this claim, starting with the fact that, well, they're assuming it will take 20 minutes for an individual to see a 2.5-minute eclipse. Second, that all individuals will go out to see it, which there's a good chance they will, but that's a separate point. Third, that the $700 million applies based on the average, that is the Labor of Statistics average, for all workers over 16, and that all workers will go out to see this, noting that there is a relatively narrow path in which the eclipse could be seen. And finally, just how much that $700 million is actually going to be in the end. $700 million over the course of a year isn't even a drop in the bucket of money being paid. It's not going to show up in anybody's books. And that's the issue here. The claim sounds amazing until you realize how little value that actually is. Even if we're operating on the premise that the smallest businesses are affected by this, there is a very good chance that those small businesses are collectively admiring the awesome astronomical event. And if they're not, there's a good chance that the management has communicated the expectations. There's no confusion about what's happening. That's all the news we have for you this week. Thank you for watching this video. If you found it interesting, consider liking, sharing, and subscribing. And please do post any comments, questions, or suggestions you have below.